Hello everyone. So today we're going to be looking at cloning, which is a very fascinating topic. One of my favorite topics to teach in biology. So let's look at the study design dot point. It says that you need to know the process and application of reproductive cloning technologies. So we're going to go through actually quite a few reproductive cloning technologies. How they're used in the real world, so their application, and also the process of how it works. What is a clone? Let's start there. So a clone is something or an organism that is genetically identical. We can also call a cell a clone if it's genetically identical to another cell, or a section of DNA or DNA in general can be a clone. So when we looked at asexual reproduction, the offspring were clones of the parents because they were genetically identical to the parents. So that's what we mean by clones. So clones can exist naturally in our world. However, reproductive cloning, so that's cloning to produce offspring, or the process of cloning to produce offspring, is artificially induced by humans. And because of this, there are ethical implications we need to consider. When clones are produced naturally, we consider this asexual reproduction. So just keep that in mind. So if a clone is produced naturally, it's asexual reproduction. If it involves human intervention, it is artificial and it's considered reproductive cloning. Here we have an example of Snuppy. Over here on the right, Snuppy is the first cloned dog. So Snuppy is the exact is genetically identical to this parent over here. Let's start with reproductive cloning technologies in plants. There are three that I want you to know. So plant tissue culturing, plant cuttings, and plant grafting. Let's look at the process of plant tissue culturing. So this is when we take cells that have the ability to differentiate, meaning stem cells, and they're taken from a plant that has grown on a highly nutrient medium. So we monitor these conditions really tightly, we regulate these conditions, and eventually those cells that we've taken develop into a tissue, which we call a callus. And this callus can then be separated into multiple portions and each grown into its own organism. So each of these portions are genetically identical to each other, and they're also genetically identical to that plant where we took those cells from. Here I have a little illustration of that. So we might take some stem cells from an adult plant, we culture that in a highly controlled environment in a lab. This develops into tissue, which we call callus. We separate the callus into separate portions, and each of these portions we can grow into a whole plant that is genetically identical to the parent. So of course, this is useful if you are trying to produce a lot of plants that are genetically identical in a very short amount of time. However, of course, this is going to be very expensive because we have to tightly control this process, and we have to do this process in a lab. Next, we have plant cuttings, and this is something that many people have done since the dawn of time. So this happens when we take a part of the plant, so maybe it's a stem, a leaf, a root, we cut it off and we replant it. Obviously, we can control the conditions and monitor the plant, and that cutting grows into its full organism. That cutting is then genetically identical to that parent plant. This is something that many people have done. You might have done this yourself, where you take a cutting of a plant, replant it into another pot, for example, and it grows into its own organism with roots and everything, and this cutting is genetically identical to the parent. Lastly, for plant cloning reproductive technologies, we're looking at plant grafting. And so this is when we take the stem, which we call the scion, of one plant, and we attach it to the rootstock, so the stem of a fully developed plant. So a scion from one plant put into the rootstock of another. This scion and rootstock will fuse together and we create a clone. So this part over here is a clone of the plant it was taken from. So the scion is genetically identical to its parent plant. Here we have a diagram for you. So we have this fully established adult plant. We cut it. So this part here is the rootstock. We now have another plant that we want to insert into this rootstock, so the scion. Maybe this is some type of apple tree that we want to grow, but we do not want to wait for it to fully develop, because apple trees can take 2 or 3 years to fully develop. So we take this scion from this apple tree we want to grow, put it into an already matured rootstock, fuse it together, and that apple tree will now grow, so that scion will grow. This scion is a clone of the parent plant that we took it from. So obviously this can save a lot of time if you are wanting to grow fruit, for example, and you do not have the 3 or so years to wait for that fruit tree to fully establish and start growing fruit. Plant grafting can actually be used very creatively. As you can see here, all of these structures you can see are a result of plant grafting, which I think is pretty cool and very creative. So what are the implications of plant cloning? Meaning, what are the effects of plant cloning? What does that mean for people and for the environment? Well, if we have genetically identical plants, this means we are hindering genetic diversity, right? So we're not increasing genetic diversity, the diversity is staying the same because we are just reproducing more of the same alleles. We're not introducing different alleles into the gene pool. And a small gene pool leaves parts of the plant population at risk of disease and the effects of environmental change. So because they do not have diversity in the population, it is really difficult for the species as a whole, or the population as a whole, to adapt to different changes. And if all the members of that population are the same and a disease comes along that affects a specific allele, for example, there is a risk of that whole population being wiped out because there is no genetic diversity. There are no other members of the population that are able to withstand that disease. Here are some fun examples of plants that are clones that you may not know of. So Cavendish bananas are actually clones. So this is the banana down here. When we eat them, this is what they look like. They are seedless. Remember, seeds are fertilized gametes. So if there are no seeds, it means there's no sexual reproduction. Therefore, a very small gene pool and susceptible to disease. Up here, this is a photo of a wild Cavendish banana. You can see the seeds. Obviously, those seeds will then go on to become more Cavendish banana plants and so on. But to produce this seedless type, we must clone those bananas. Therefore, our banana population is actually at high risk of being wiped out if a disease comes along that affects the bananas, and this has happened in the past years. Granny Smith apples are also clones. Apples are very notorious for not being able to be grown from seeds because there's so many mutations that occur in apples. So a lot of apple farmers will use plant grafting to ensure that the same type of apple is grown. A very interesting story about Granny Smith apples, which I will tell you in class. But Granny Smith apples are another type of fruit that are clones, and we eat them all the time. So are navel oranges. Again, they're seedless, and so they have to be cloned in order to be produced, and they're produced via plant grafting. Have a think about what this means for our food sources. If we have all these plants that are produced via cloning, and so all the plants are genetically identical, what does that mean for our food sources? Now let's look at some reproductive cloning technologies in animals. There are 2 types that I want you to know. So we're looking at somatic cell nuclear transfer and embryo splitting. Both are very, very interesting. Let's start with somatic cell nuclear transfer. Let's break down this name firstly. So somatic, we know, refers to non-reproductive cells, or somatic cell is a non-reproductive cell. Nuclear refers to a nucleus, and so we are transferring the nucleus of a somatic cell in this reproductive cloning technology. The process of somatic cell nuclear transfer, or SCNT, is 4 steps. So we have enucleation. So we take the nucleus of a donor egg cell and we remove it, but we keep that empty egg cell, so that enucleated cell. We then have extraction. So we remove the nucleus of a somatic cell and we keep that nucleus. So we have an enucleated egg cell and a nucleus of a somatic cell. Now we have insertion. So we insert that nucleus into the enucleated egg cell and we allow for development. So after some treatment to make the cell begin dividing, that embryo is then transplanted into a surrogate, which then will develop the embryo. So a surrogate is an animal that has a fertilized egg put into its uterus to then develop that embryo into an offspring and give birth to that offspring. So the surrogate is not the biological parent. It is just developing that offspring in its uterus. Dolly was the first sheep, or the first animal, that was created using somatic cell nuclear transfer in 1996. However, Dolly died 6 years after that. But the cloning of Dolly was a huge development in terms of cloning reproduction, or reproductive cloning technologies, and it was very, very interesting, and Dolly's legacy lives on. So how did Dolly come to be? Well, we took the egg cell from the egg donor. So this was the Scottish blackface sheep. So we took that egg cell, we removed the nucleus. Now we have a Finn Dorset sheep, so a different type of sheep, and we took some mammary cells, and from one of those cells we took the nucleus and we fused it with this empty cell. So now this is a fertilized egg. Sorry, it is an egg rather that has 2 sets of chromosomes from this Finn Dorset sheep up here. So this is the nuclear donor. That is then developed in a lab, and that blastocyst, which is a developed embryo, is put into the surrogate ewe, and this ewe, which is a female sheep, gave birth to Dolly. Now Dolly is a clone of this nuclear donor up here, right? However, to make Dolly, there was 1, 2, 3 sheep involved to make this one clone. Very interesting. This video shows you the process of how
This video was prepared by my colleagues Dieter Egli and Kevin pipette, and then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you 2, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again, a little hole, and the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transfer.
Here we have some other examples of animals that have been cloned. So we have Cece the cat that was cloned in 2001 and lived for 18 years. Barbra Streisand, if you know her, she's a famous singer and actress. She loved her dog Scarlett so much that she cloned them. Sorry, she loved her dog Samantha so much. So Samantha was the original dog, and she loved Samantha so much that she made multiple clones of Samantha. This costs tens of thousands of dollars. So really only someone with a lot of money would be able to do this multiple times over for their own pets. So now the big question that everyone always asks, can humans be cloned using somatic cell nuclear transfer? Theoretically, yes. However, it's illegal, right? So some scientists have tried cloning human embryos with no success though. It's actually quite difficult to extract the nucleus from a human cell and primate cells because of the centrioles and the position of the centrioles. Usually when you're removing that nucleus, the centrioles come with them. And if you remember back to mitosis and meiosis, the centrioles are very important for the cell to be able to divide. And so without the centrioles, division cannot occur. And therefore, we're not usually able to do this with human embryos. But theoretically it is possible. Of course, illegal in most countries, and you can think of the ethical reasons why. Somatic cell nuclear transfer, however, can be used for therapeutic cloning. So this is cloning for medical therapy reasons. So we can use it to develop stem cells with the same DNA as the donor cell. However, we have no evidence of this occurring. But this is not a reproductive cloning technology. This is used for medical reasons to treat diseases, for example. So we can also use somatic cell nuclear transfer to study diseases and develop new treatments. So for example, we can grow healthy tissue to replace damaged tissue, or we can test new drugs on tissue grown from stem cells. The process is still the same. So we have an embryo with a donor nucleus and develops for 5 days. The stem cells are then collected, and then we destroy the embryo. However, those cells all come from that same parent. They're all clones. And this is really useful when we're trying to study different diseases and treatments. And lastly, we have embryo splitting. So this is the embryo development of a human. You don't need to know these different stages. I just wanted to show you how you go from one fertilized egg in day 1 to 2 that divide via mitosis. They're dividing again and again and again and again. So you go from fertilized egg, 2 cells to 4 cells to 8 cells, 16, and so on till you have a fetus. This is the same for cows. You have one cell that divides into 2, into 4, and so on till you have a fully grown organism. Embryo splitting then is when you take one of those eggs that are fertilized, usually via IVF. So IVF is when it's fertilized in vitro, so in a lab. And once that embryo begins to develop, you split it once it gets to that 8 cell stage. So if you think back to the past 2 slides that we looked at, once it gets to that 8 cell stage, you split it. At this stage, those cells are pluripotent. So they're stem cells. They're able to differentiate into many different types of cells. And each portion of that embryo that you split will continue to develop into a full organism. We take those different separated embryos now and we insert them into a surrogate or surrogates, and they develop inside the surrogate, and all the individuals, all the offspring that are produced, they are all genetically identical to each other. So this is where it differs from the other techniques. Whereas in the other techniques, we were making clones of a parent. However, in this technique, the offspring aren't clones of the parent because they've been fertilized. So we have the sperm and the egg. So we have those 2 different, that genetic recombination in that fertilized egg. And then once we have this embryo that's developed at the 8 cell stage, we then split it. And so all those portions that we've split it into will grow into organisms. So say we split it into 4 different organisms. those 4 organisms will all be genetically identical to each other. So they are clones of each other rather than clones of the parent. So that's the big difference that I want you to know. So this is one way we might do that. So we have this diagram, it goes a little bit weird, it goes down here, then across and down. So we've got a embryo that has developed to the 1, 2, 3, 4, 5, 6, 7, 8 cell stage. And now we have an empty egg cell. We take out some of those cells from that developed embryo and we put it into the empty egg cell. So now we have 2 embryos with cells in them that are genetically identical to each other. Another way that we can do this is literally cutting a cell in half. We got a developed embryo here, very closely and precisely we will split it in half, and these 2 halves will develop into 2 different embryos that are genetically identical to each other. In here I have a video again illustrating this process of embryo splitting. So we have a developed embryo at the 8-cell stage. Some of these cells are being removed from that embryo and they will go on to be grown into another embryo that is genetically identical. Here's a diagram of that. So we have our original shape that is in the embryo stage. We separate those cells, they are grown, probably in a lab, developed, put into a surrogate. So we put each of these these embryos into a surrogate. That embryo develops into the offspring, and these offspring are all genetically identical to each other. So let's look at the agricultural uses of embryo splitting. So they're usually used to clone highly favorable livestock. So for example, we might take a fertilized egg from a cattle with favorable traits such as high muscle mass, high milk production, whatever we're looking for, whatever we want to make money off. And we split that embryo into multiple offspring, and each of these offspring will have those favorable traits that we can then make money off. For example, this is how important this is to agriculture. This is Missy the cow. Missy the cow has such good genetics that she was sold for $1.2 million in Canada because her traits were so favorable and they produced a lot of money, obviously for her owners, that she was worth $1.2 million. So even though this can make you a lot of money, it is still very expensive because you need to harvest the eggs and the sperm, you need the time and the money for IVF, so fertilizing those eggs in a lab. Often it requires multiple attempts to be successful, so not every egg that you fertilize in a lab is going to grow into an embryo, and we have to use surrogates, so there are multiple animals that are involved with this. However, we can freeze those embryos with those favorable traits for later use. Again, that is expensive because you're paying to freeze them. And lastly, can we use embryo splitting in humans? It has been successfully done and as a possible use as a treatment for infertility, but it's not currently used. Again, it hinders genetic diversity though. So we're going to have the offspring are all going to be clones of each other. Of course, there are ethical considerations because cloning of humans is not legal. So I'm going to leave you here. I couldn't find a good meme for cloning. So if you find one, send it to me. But I'm going to leave you with all of this. Please write down your questions and bring them to class, and I will see you then.